Okay, record. Okay. All right, you guys, let me share my screen and we're gonna go ahead and get to it. Share. Am I sharing? Okay. All right. Am I sharing, you guys? Yeah.
Yes, it's loading.
Okay, cool. So I'll get started. So basically for let me move this guy. It's so annoying. Okay. So basically for the experiment that I'm working on with Laura right now, this is what I'm doing. We took a picture of our cells. So these are tip cells, and it's just a bright field image at 2x. So we capture the entire like droplet. And so the very first thing you want to do for Weka, if you notice, this is a 16-bit image, and that's too heavy. That's too big. Requires too much memory to save. So you just go to image type and 8-bit. So we can make the image smaller and easier for Weka to do stuff with. So now we're going to create a classifier. You go to plugins, segmentation, and then you go to, oh, hang on. I want to make sure Ariel's here. Ariel, are you here?
Yes, I am taking notes. Thank you.
Cool. So then we go to trainable Weka segmentation. If you need me to slow down or if you need me to repeat anything, just let me know. Okay, so then right here we have our Weka, and we have our menu on the left-hand side. So here is train classifier. Once you're ready for it to train, load classifier, load data, create new class, settings. So I don't know how to use everything with Weka, but I'll teach you what I know. So here you go to settings, and the only thing I do is I select the class. So for example, I'm going to put fibrin because that circle in the middle is fibrin with cells. And then the other class that I'm going to have is, I'm going to say background. And then I'm just going to press OK. Okay. All right, so now right here on my labels, we have add fibrin, add to fibrin, and add to background. So you can also create a new class and it'll create a new label, but I've never been able to get Weka to work with 3 classes. I've only been able to get it to work with 2. So that's for you if you want to play with it. Now, the next thing we want to do is we want to tell Weka what is what, like what is our fibrin and what is our background. We want to show it. So we're going to go ahead and use these little buttons that are on the Weka menu. Now, the more surface area that you take, for example, I'm going to take this circle and I'm going to go ahead and circle that little blob right there. And I'm going to say that's background. So I kind of took a lot of surface area for that. And that's going to make this classifier more heavy and more slow. So I try to stick to this little guy right here. And then you'll see I'm going to zoom in. And then so I try to stick to that guy right there. And then I just kind of like outline like what I consider to be fibrin. So all this is fibrin. So add to fibrin. And then I'll grab some on this side over here. We'll say, oh, this is all fibrin. So add to fibrin, and then you can zoom out a little bit, and then you also want to make sure you get every single corner, especially if you notice that there's like, this is really bright right here and that's dark. So you want to make sure you get samples of both, like that right there. And then this corner is background, and then this corner is background. And then this right here is like my sample fibrin. Okay, so now what would happen if I mess up? So I'm going to show you a mistake real quick. Let's say that I'm trying to go, whoops. Okay, so let's say that I'm trying to go through right here. And then, oh no, I accidentally came out of the fibrin circle. So now Weka is going to be very confused and it's not going to be able to train properly if I leave it like that, but I'm going to add it to Fibrin. So I just added it. So that's not good. Weka is not going to be, it's going to be so confused. The algorithm is going to be so messed up. So you definitely want to delete that. All you got to do is double click on this guy and it should disappear. It's just very slow right now, but you see that it just disappeared. Okay. So then when we're done, let me do one more. Okay, so now I'm going to say, okay, I think I provided enough information for Weka to know that all I want is this circle. That is all I want in life. So I'm going to say train classifier. So now Weka is going to, we're trying to create a model. So our model is the classifier. So Weka is going to try to create that based on what we taught it, and we're going to see if it works. So I'm going to stop recording for now because it takes a little while. Let's see. And then my computer's extra slow because I'm on Zoom and I'm sharing the screen, so just bear with me. I don't know how to pause the recording because the menu's not showing up because it's too slow. Fiji takes a lot of computational power, so just know that. Does anyone have any questions right now? I think someone's talking, but you're on mute. Zoom recording. Okay, so that took about 20 minutes and it's done. You know that it's done when all of these buttons become active again. So everything that was marked green was my background. That's what Weka learned, and it said, okay, from what we learned, we think that all of this green stuff is background. And from what it learned, it thinks that all of this red is my fibrin. So it did a wonderful job. Now, do I think this classifier is perfect? No, I don't think it's perfect. I would work on it a little bit more. So like I would go in and teach Weka that this right here is all still, like I still want it. It's all fibrin. And then you hit train classifier again, and then you have to wait again. So that's the secret to getting a really good classifier. You need to go through all the details and make sure that it's perfect, that it learned everything perfectly. So for you, Ariel, since you want certain cells that are at the top layer, you said that was like the left-hand side, you would only go in and select those, and then you would make sure that everything on the right is green, because it's background. So as long as you teach it properly, you're going to have beautiful results. So then you go to save classifier. Once you're happy with it, you go save classifier. I like to save it in the location where that data is at. So like wherever you have those pictures in that folder, go in there, create a new folder inside, a nested folder that says classifiers. And then that's where you're going to keep all of your classifiers for that experiment. Now, whatever you label it, you're going to say dot model. So label it something that describes the picture or the experiment. That's what I do. Label the magnification. So I always do 2x or 10x or whatever. And then dot model. Okay, as long as it says dot model, it'll save it properly as a classifier. If you don't put dot model, it's not going to work and you're going to have to start over. So I saved mine yesterday. So I have like a good one that I want to use. So I'm not going to save this one. But what you do next is get probability. And this is where you have your segmented image. It's going to give you like a segmented image, and that's what you save as a .tiff. And that image is the one that you can go ahead and process with everything and anything you want, Ariel. So you would have another folder that says processed Weka images or something, or segmented images. And those guys, all of those images in that folder are going to be ready to go to MATLAB or whatever it is you do. Okay.
Mhm.
And this one,
Got it. That makes
and you're supposed to be able to automate Weka. I haven't figured out how to. I know how to automate like one way, so I'll teach you guys that later. But you're supposed to be able to automate it also in a coding way, but I haven't been successful at it so far.
okay.
Okay, it's almost done. So you'll see the segmented results. There we go. You see how beautiful that is?
Oh yeah. I see it.
Because all we want is we want to be able to quantify like these guys. So for you, you're going to want to be able to quantify your little beads, right?
Mm,
So
yeah.
your beads are going to be white and then the background is going to be black. And then since you have a movie, your little beads are going to move like
Mhm.
from left to right. Like I'll show you here because here I had my beads. I think they weren't beads, but they were like um cells, but they were like moving. Let's see if we could. Yeah, right here. You'll see. Yours is going to be kind of like this. You see that how they're moving?
Yeah, exactly. Yeah.
So your beads are going to
Mm,
be doing that. So this is an image that this is a Z stack that I classified. So Weka went in and got rid of all the junk that I didn't want, and it only kept the beautiful fluorescence that I wanted. And then it it segmented the entire image. So then when I put it into my cell tracking, and because this image was already calibrated, it went ahead and got distances for me, it got velocities, it got so many statistics that I haven't even gone through. So that's what you're going to do. With this process right here, it only takes about 15 minutes per Z stack. So I think I really like it. But you can also try it your way and see if you get similar numbers. That would be interesting.
okay. Yeah.
But yeah, so the last little thing is that this guy right here is it's two images and we can't do any kind of analysis with that. So I always like image. So at this point I have like a code where I just go from stack to images, and then I only save this guy, this one. I just save it as a TIFF so that I can process it later. And then the last thing I wanted to show you guys is the classifier is right here. So you can just apply classifier. And now you can select any other image that's similar.
Yeah.
And then you just press open and you say get probability. And then bam, it'll apply whatever it learned here to any other image.
Okay.
Okay. Oh, and then if you had Weka closed and you want to load up the classifier that you saved, then you just go to load classifier and then it'll ask you what image you want to use it on and you just go and select it. Easy?
Yep. That looks doable.
Awesome. Does anyone have any questions? Someone's talking, but you're muted.
That's the second time you said that